Right. Welcome to another episode of Summertime Science. I thought for today's episode, being summertime, we should go to possibly the epitome of summer, which is Hawaii. I'm celebrating in my own way because this shirt is as close as I'm going to get to Hawaii. However, I have the pleasure of talking to Dr. Chris Hobbs, who is in Hawaii at the moment. That isn't actually one of those fancy animated backgrounds behind him. That is actually the coast of Hawaii. So welcome to Summertime Science, Chris, and thank you for spending some time with us.
Oh, thank you very much for having me. It's great to be able to talk with you and talk a little bit about my research and how I got here, really.
Yeah, thank you. So what I'd like to do is just get an idea of, firstly, how you came to university and sort of what your experiences were. And then I'd like to get straight on and talk about your research. So just to start, I mean, I know you from Christchurch. That was where I first came across you. How did you come to be at Christchurch?
So when I was at primary school and stuff, I always knew I wanted to become a scientist or really study science. And was very passionate about it and worked hard for it. But during my A-level exams, I came down with a nasty illness and was withdrawn from my exam. So I actually didn't get any A-levels. And I was planning to retake the last year of my A-levels when Canterbury Christchurch gave me an alternative offer of actually going in, instead of in first year, going in through the foundation year at Christchurch. They were like, hey, you may not have done your exams well, but we like your style anyway. And so it was a fantastic opportunity to get to university, study for just an extra year, and get my feet underneath me at the university. And it was an incredible opportunity. I loved every minute of doing my foundation year there, and it really grounded me and helped me settle in and move forward with my studies. It was an incredible opportunity.
Okay, just in case there's anyone watching who's thinking about going to university, what would your advice be for getting the most out of university?
Honestly, my experience is you try everything and anything. Volunteer for what you can. If there is a position coming up, like a student representative position, give it a go. Try out something new wherever you can. It makes a huge difference to you as a person and also to your academic experience. Canterbury Christchurch, when I was there and still does, offers a lot of lab-based internships during the summer terms there. Try and go for those as much as you can from any year of study, from foundation to final year. They are an incredible opportunity that give you such varied experience in the lab that not only helps you for that specific subject, but will also help you for your greater studies and your academic development. You'll learn how to concisely put together a lab notebook, which is very important when you're doing this sort of work, and give you that experience early on that you can build from there, and it will greatly help you on later on. And also, more importantly, it gets you to do some cool science that you have never done before, working with some of the biggest experts in their fields at the cutting edge of research, and it's incredible. So one other thing that I did when I was at university is I went on an Erasmus year. So I studied abroad in Germany for my second year. So I don't know how that currently is going to happen with the current situation we're in, but if you can get an opportunity to do that, studying abroad it opens your eyes to a lot of different cultural aspects and makes you question how you act and the way you learn, because all university systems in different countries are different, and it gives you that opportunity to push your boundaries. So I would say do that, and also explore local companies in the area. There are so many science companies in Kent that Christchurch has great links with, and your lecturers will know those members of staff. And so when everything opens up again, if you can, volunteer with companies as well. We have multiple graduates from Christchurch who now work at the companies they either interned it with or did their dissertation research with. So there's so many opportunities, and you need to take them.
No, that sounds really good. How did you personally balance work and your university work with sort of enjoying life? Because I mean, some people think you need to kind of go one way or the other.
It's a balancing act. It is very tough. I can't. There's no way to say it any other way. You'll have to find your own balance, and it may take some time. When I started, I leant very heavily towards the fun side, and then got my footing a bit better and brought it back. Not a lot, but I brought it back. And so you can't just kill yourself with the research and with studying, because you won't develop the social skills you need to be able to interact in the workplaces later on. You need to have both. So I joined a few societies. I was heavily involved in the fencing society at Christchurch. I was on the team, and that was a fantastic experience. I learned a lot, drank a lot, and I interacted with people on so many different courses that really sort of broadened my horizons in many ways. You can't just let it take over. Also, try and when you've got assignments and stuff, try and get them done early, because then that's not constantly nagging at the back of your mind whenever you are having fun. If you get it out of the way early, you feel a bit more of a release and can actually enjoy yourself much better.
Okay, I mean, we're going to talk about Hawaii in a moment, but I'm just wondering, fast forwarding to now, what do you do at the moment for fun and entertainment and to sort of relax away from work?
So actually, I have taken up surfing whilst I'm in Hawaii, because I mean, if you go to Hawaii and don't surf, you're doing something wrong. So the waves are actually pretty good today. I was out there earlier this morning. It was really good. So I try and surf about 2 to 3 times a week. I joined a running club whilst I was out here called Run, Drink, Repeat.
Nice.
They're a great bunch of guys and girls. All come together, start I run at a pub, run a 4-mile circuit, finish back at the pub, have a few drinks, then possibly have some more. And so I always try and find something that's relatively healthy to do, that I can do in a very social aspect. I mean, less social now, obviously. There is obvious social distancing rules here because this is very important. But yeah, that's what I've mostly been doing here is got a couple of surfboards and try and get out whenever I can.
Okay. Right, so you've done your degree. What on earth possessed you to do a PhD?
Pure unbridled insanity, I believe, is mostly what did that.
Yeah.
I'm not really sure what really. There's no one impetus that made me want to do that. I enjoyed doing my dissertation research, which I did on mussels and their growth in response to algae on their shells. And so that was a lot of fun, of being on Ramsgate Beach at 3 o'clock in the morning because of the tides and the sleet in February. And I'm very happy to say I've somewhat upgraded to a slightly nicer beach since then. But I think one of the main things that really got me interested in the research, and especially sort of conservation and ecology research, was the 3rd year ecology field course offered by Christchurch. So this was a course where we went to Wales on a residential field course, and it was spectacular. It was hands down the best part of my degree, by far. It completely changed my life. In the course, you get to design, carry out, and present a research project you've designed, that your group has come together and gone, this is what we want to explore. And you get to get all that data. You get to work with experts who give you a load of information and help with it. And also, you're in the mountains of Wales. I mean, there's just some of the most spectacular landscapes in the UK. It really opened my eyes to the fun and possibilities of further study through research. And since then, I just kind of ran with it.
Okay, so I mean, you've solves your idea of how much you enjoy science and you liked ecology. Why snails, though? You might have to explain roughly what you did for your PhD as well.
Okay, so for my PhD, I studied an endangered species of freshwater snail called Segmentina nitida. So it's a small, about 5 mm snail found in sort of like drainage ditches and sort of stagnant waters. And it's
already people looking at this are thinking, I've got to come and study the snail.
Yeah,
you get to drainage ditches and you get to search them for tiny snails.
Tiny snails, often near winter, in the freezing cold.
Beautiful.
Occasionally swamps. It's perfect. So it's with this species, it's thought to be across Europe, and in the UK, it's declining quite badly because of anthropogenic means. So eutrophication from fertilizers, mechanical clearage of ditches, and habitat fragmentation. So that's what I studied. And I've always been fascinated with conservation, and possibly a little bit naively, I wanted to make a bit of a difference with my research. I'm not saying that's the biggest thing about it, and it's not something you should always strive for, but it's something I always see.
This little worm in the back of your head. Oh, it'd be great if this changed, in your case, the world of ditch management or something like that.
One tiny snail. With this one, with many invertebrates, it's not just that one tiny snail. They're very important in many ecosystem functions. They help with nutrient cycling, cleaning of detritus, and just providing parts of the food chain that some other macrovertebrates may not provide as much. And invertebrates, snails, they have made up the majority of known extinctions since the 1500s. Like, it dwarfs vertebrate extinctions by far. They're so hundreds of species have been documented of going extinct, and many that we have no idea about. So they're a very high extinction risk, but they get a lot less attention because they're regarded as icky. They're not charismatic like some of the larger things. Like everyone loves tigers, everyone loves pandas, got to save those. But invertebrates are cared about less, but they offer a lot. And so I got into that mindset of conservation, getting away from sort of like what's regarded as keystone species, ones that charismatic and then money can sort of trickle down to the others, but actually focusing on those others.
Okay.
And then
so we've got this stage where you're looking at this snail. You've got an idea. It's rare and endangered in the UK. You're going to do something to help it with its conservation. Did your while you were doing your research, did you head in a kind of a straight direction towards conservation of Segmentina, or did anything else happen?
Lots of other things happened. It's a very fluid way of doing this. You start a PhD, you start a master's, whatever. You think it's going in one direction, then a curveball will happen, and suddenly it will completely change, and you're going nowhere near that direction. So I was going to just look at, in the UK, ways to whereabouts it is first, establish like geographical maps, then try and do captive rearing, and then break out to reintroductions and see what happened there. And that did not happen. So the thing about endangered species is they're often endangered for a reason. And so replenishing them can be quite difficult. And so, especially in like quite complex habitats like drainage ditches or similar, it's really, really hard to recreate that in a lab, unsurprisingly. And so in the lab, they didn't respond too well to captive rearing. So quite a few died. So that area of my research then stopped. And so I realized that before you can reintroduce a species to a location, you need to understand its genetics. You need to understand population genetics, the diversity within it, and a sort of like a meta community in its whole range. So I went to explore the genetics of it across Europe, because it's meant to be such a widespread species. Could we take individuals from other populations in Europe and translocate them back to the UK, expanding their range? And through this, I actually found an amazing thing is that there is not one species. So Segmentina nitida, as we know it as one species. The genetic data and morphological data combined indicate that there are actually at least 2 species in Europe. So I went to Germany, Poland, Sweden, and the UK, surveyed there, and it actually looks like Germany has a mixture of both species, and Poland and Sweden, Poland especially, only has one species. The UK has the other species, and there's one population in Sweden that has the same species as the UK, but the rest are all the same as the ones in Poland.
Right.
So that has a huge impact on the conservation of the species. If you have a species that you consider endangered and suddenly its range is slashed in half, it's more endangered. So we need to have quite strong management protocols to try and protect this. And also management changes depending on how it is. So it's very important to have accurate taxonomy, or the association of one species name to one organism and another one to another one for conservation. If you don't know what you're conserving, you can't conserve it. So taxonomy is one of the most important things in scientific research for conservation, in my opinion, that we can do. So my research ended up being geared somewhat towards that.
So what did you do to find these different species? Because presumably the reason why they were identified as Segmentina nitida is because the morphology, the shapes of the animals and their shells is very similar.
Oh, they're non-indistinct is the way to say that. They are very, very morphologically similar. So doing it by eye is quite difficult because there's just minimal changes in the aperture, where the hole where the snail comes out of. There's very small differences there. So what I utilized is a method called geometric morphometrics. So this is a landmark-based method of shape analysis. So you take your pictures of your organism, and then you define areas on that shell which are present in every single individual in your data set. So let's say,
if you've got a snail shell, you'd say have it on its end, and you'd have top of the aperture, bottom of the aperture, sides, and then top of the spire, that sort of thing. And each one of those landmarks is going to be the same for every shell. Exactly.
So it's a one-on-one thing. So they have to be definable locations. This landmark can only be placed at this specific location. The widest point of a shell, the meeting of 2 structures. You have to have it so it's all homologous.
Okay.
So then you align them to each other using software. They align and scale them. So you're looking purely at shape. There's no differences in size. Everything's the same size, same orientation, and they're all just completely transposed over each other.
Okay.
Then you look at how those landmarks are different between all individuals and between defined groups. And so you can actually put this, so you can do statistical tests, and you can do modeling that means you can discriminate between the groups and assign individuals to a certain group.
Okay.
And how much accuracy that model can do. So when I first took these shells and then just assigned the shells to the country we found them in. So we grouped them by country. The model could only say which shell belonged to which country about like 49% of the time, which is terrible, obviously. Yeah. But then when I included my genetic data and said, hey, there's these 2 species, it jumped to nearly 90% of getting it to the correct species.
Right, okay.
So this is a method called, this is a form of integrative taxonomy. So this is putting together multiple lines of evidence together to be able to accurately say, this is one species, this is another. So you can add anatomy as well, radula. So snails feed by rasping sort of their teeth against a surface.
Yeah.
Rubbing it together. And so you can take scanning electron microscopy images of that, and that will also add evidence to taxonomy, to description of the species. There are it's a multifaceted way of understanding species. And so this genetics and morphology are two steps in that to be able to say this is exactly a species.
Right, so you've taken your geometric morphometric data, which has identified essentially two groups. You've taken your genetic data, which has identified two groups, and then you've looked at the two sets of data, and they've identified pretty much the same two groups. So now you've got a very good idea. You've got these two separate species. Well, that's fantastic. And so I think we're now getting towards the end of your PhD. That was quick.
It seems a lot quicker now.
And towards the end of your PhD, you're starting to look around for somewhere else to go. How did you end up in Hawaii?
So in 2017, the chair of my supervisory group, Professor George Desart, sent me a link to a conference in America saying, hey, I think you should go for this. When I went on to this same group, it pretty much changed my career. I think you should go for it. Give it a go. So I applied. I submitted an abstract, got awarded a grant to go to the museum. So it was based in Delaware. So I went to the Delaware Museum of Natural History, where they actually had segmentina samples, which was amazing. So not from America, they've been collected in areas of Europe and then brought over. So I went to this conference. I flew out alone because my supervisors wouldn't come with me for some reason. Don't know why. Thanks, Phil. So I ended up going out a couple of days before the conference because of the time zone issues and flight times, I missed the first half a day or so of the conference, so I didn't want that. So I turned up, and there's a pre-meeting session with, on digitizing museum collections and how they can bring the back of house stuff out forwards for either research or for engagement with the public. Really cool, and that's kind of something I like to do here. And this essentially had every major museum in America. It had their collections managers or curators for the mollusca department there, and me.
So all the big movers and shakers in the world of mollusks in America.
And me.
And you. Okay.
Which was slightly daunting until the first night after the talks. And believe me, these guys could drink. We had a social evening, we had multiple social evenings, but this first one had an absolutely amazing dinner, drinks afterwards, and Jesus, I have never been that hungover since, I don't think. And then during that night and throughout the rest of the conference, I just talked to everyone. Chatted to people, talked about their research, went to the talks, engaged with them, gave my talk, which everyone really liked.
Yeah.
And then towards the end of it, I was approached by who is now my boss, Dr. Ken Hayes. He sort of approached me and said, hey, I like your research, I like what you do, I like how you interact with people. You considered postdoc, can you finish your PhD by then? And so I was just coming up to my third year out of a 4-year PhD, and I was like, there is absolutely no way I can finish a year early. I would be abandoning so much research. Yeah. So I said, unfortunately, no. So I didn't get that postdoc. And so I was a little bit bummed by it, but these things happen.
Yeah.
So I declined, and then the next year there was another American Malacological Society conference, one that the same with Delaware, but this time it's based out here in Hawaii. And did socializing, did the engagement, the whole lot, gave a nice presentation, and then Ken approached me again and said, hey, some more money's come up. January, February 2019. How'd you feel about it? And naturally, I accepted.
Really, that's really useful. Now, we've now found you in Hawaii, where you are now. What do you, apart from sitting on balconies overlooking the sea, what are you doing?
Not much, really. That's pretty much what I do every day. I don't do anything, really. No, I joke. So I'm based in what's known as the Pacific Center for Molecular Biology, PCMB, at the Bernice Pauahi Bishop Museum in Honolulu. So I work on quite a lot of different projects, but my main focus, my main research interest is utilizing museum collections primarily for conservation. So natural history museums, obviously, they're ubiquitous. They're all around the world, and they have fantastic back-of-house collections. And so for snails, a lot of the collections are primarily dry lots. They're empty shells. And so you can't get, it's hard to get genetic data out of them. And you can find it's like a single species, individuals going back often over 100 years.
Okay.
And so, for example, the Bishop Museum I work at, we have one of the most comprehensive land snail collections, Pacific land snail collections in the world. We have over 4 million specimens in there, catalogued. So we have at least another 2 million on top of that. We have nearly every species that is found in Hawaii. So on a related note to that, Hawaii is incredible. Hawaii, for evolutionary senses, is, especially for snails, is spectacular. As one of the best land snail radiations in the world, where they've colonized at one part and then spread through and speciated as they go through the island chain. So as far as we know, there are at least 750 described species of snail just in Hawaii.
Oh.
And a lot undescribed. And 99% of that 750 are only found in Hawaii. They are endemic. They are found nowhere else in the world. But also Hawaii is somewhat of a extinction hotspot. I think it's regarded as like the extinction capital of the world, which, I mean, if you've got to be known for something, that's probably something you don't want to be known for. And so we have problems with invasive species, climate change, and changes of land use that have made it very dangerous for these species, and a lot of them are dying out. So that's why conservation is such a key piece of research here.
Okay. Could you just give me an idea of these endemics, this huge range of endemic species, how distinct they are from other populations? I mean, are there different species from families that you find in other places, or are there kind of distinct families or that sort of thing?
So there are members of these families that are found throughout the Pacific. So the families aren't always fully endemic. A lot of them are, but for the idea of it, there are certain species that are restricted to like a single mountain range or a single mountain top.
Wow.
So they don't disperse very far, which has caused this radiation of species. And so you'll find pockets of these species. But you can find members of these families elsewhere in the world.
Okay.
Many aren't.
Okay. So, what particular species are you focusing on? Because I suppose you can't look at all of them.
No. So I primarily work within a family called the Acatinellidae. So they contain trees, the Oahu tree snails, the Hawaiian tree snails. So there's certain subfamilies within that. And I work on a genus called Auriculella, which is another tree snail. So it's called Auriculella. So auricle, sort of like an ear. So their aperture is slightly ear-shaped.
Right. That's easy to remember. Auriculella. Okay.
Yeah. And so a lot of these species are becoming extinct. They're very pretty shells, which I'll show in a little while. And so for that project that I work on, I work with the museum collection. So we have some specimens from the last like 5 to 10 years that my research group has collected that we have genetic data for. Okay. So I work primarily on 4 species of auriculella that are morphologically indistinct. They basically look the same, but through genetic data, some anatomy, and also through the shells are just fairly distinct, distinct enough that with a large amount of taxonomic knowledge and working with those species a lot, you can tell them apart. But what my research is doing is using geometric morphometrics, which I talked about earlier, to provide a rapid way with minimal training to be able to delimit these species, to be able to tell them apart.
Okay. And
also look at how their shape has changed through time. This is the utilization part I was talking about, is that we can basically either map some form like evolutionary pressures, or we can look at how diversity has changed through time using the morphology as a proxy for genetics in some ways. Okay. So if there's a lot of shape variation 100 years ago, and there's next to no shape variation now, and their genetics now also isn't very varied, we can sort of imply that there's been a contraction in morphology and likely genetics that makes this species much more at risk for extinction. And so we can map this morphological disparity like through time, like per decade, as a time series, and see what has changed.
And this is because the collections, by their nature, are of the shell without the genetic contents.
Yeah. So okay. That also kind of brings me on to my other piece of research. So my other piece of research is still ongoing. It's been very tough to do, but I'm looking at extracting DNA from shells, so we can actually utilize the collection even better. So when snails grow, they start off as a teeny tiny little shell. As they grow by an accretionary means. So they just tack on a new layer onto their shell each time, as they go on, and the shell gets bigger, curls out, the spiral. Yeah. So even if it's like a tall spired one, or a discoid shell, a helicoid spiral, they will still grow it on like that. It's still a spiral. It's just how tall it is the difference. And so as it does that, pieces of its mantle of the snail may get caught in the shell and trapped within the micro layers of that shell. Ow. So even after you completely remove the shell, the snail out, the body out, you can still have pieces of this DNA. It's highly fragmented, teeny tiny pieces of DNA. And so what I do is crush up the shell. So at the moment, I'm just working with invasive species and nothing from the main collection that might be valuable. Yeah. But as I said, we've got over 4 million. So there are some shells that we can sacrifice to the science gods to be able to try and get this DNA out.
Okay.
And so what I do is then use amplify these small fragments for a gene called cytochrome oxidase subunit 1, cytochrome 2 oxidase subunit 1, I do apologize, which is known as sort of like the barcoding gene. It's one that we've amplified for many species, thousands of species across the world, that we can compare to and have it like a genetic barcode for it. And so I amplified these small fragments into this, and then we can assemble the gene back together. And then that allows us to have genetic data from an individual 100 years ago. So giving it a looking glass into the past, and sort of saying, so some people say like, and how does that help us conserve something? You know what it looked like 100 years ago, but that gives us a genetic baseline. So when they weren't endangered and they weren't in tiny little populations, this is their genetic diversity, potentially huge genetic diversity. We now look at now, they've got a tiny genetic diversity. They've lost a lot of it, they're at high extinction risk. But maybe if they had next to no genetic diversity then, and they have very little now, it's not quite as much of a huge decrease. So it may be less of an extinction risk for them. So you would prioritize the species that have lost the most genetic data,
Yeah,
the most genetic diversity. And so it's informing management. You can't conserve something if you don't understand what it is. So taxonomy is a part of that, but also its baseline genetic diversity is a huge part of that.
yeah, that's really interesting. One thing you did tell me in a previous conversation that I found quite exciting. Apparently, you've still got not just the shells, but whole animals that were found and preserved by Captain Cook. Is that true?
We do. We have some from Captain Cook, but we've also got ones from later on, from like an early turn of the century, that are preserved in pineapple ethanol.
Whoa.
So many museum collections around that time used formalin as a preserving agent. So that cross-links the proteins, makes it's incredible for stabilization. They ain't going to rot. They're not going to degrade in any way, but it's super hard to get genetic data out of it.
Right.
It's becoming more possible, but it's still evolving research there. But these ones, they just took pineapple ethanol, which doesn't do that. It preserves it, but it still means we can possibly get genetic data out of them. So we have these huge cabinets full of alcohol and snails, not just alcohol, that we could potentially use. And we have still, like, some of them are dissected, so we have their anatomy there from the research that was done early on in the museum's history. Super cool stuff.
Yeah, so I suppose at some point you might be able to get hold of them and do some genetic analysis.
It's definitely a possibility.
Excellent. Oh, that's really exciting. Right, well, thank you very much, Dr. Chris Hobbs, for talking to us. And I hope people and anyone who's watching this has enjoyed a little insight into, firstly, a kind of an early scientific career, and also into the research that you're carrying out. Thank you very much.
Well, thank you. Thank you for having me.